level of construction, all cells possess a few fundamental components. These include cytoplasm, a gel-like substance composed of water and dissolved chemicals needed for growth, which is contained within a plasma membrane, also called a cell membrane or cytoplasmic membrane, one or more chromosomes, which contain the genetic blueprints of the cell, and ribosomes, organelles used for the production of proteins. Beyond these basic components, cells can vary greatly between organisms, even within the same multicellular organism. The two largest categories of cells, prokaryotic cells and eukaryotic cells, are defined by major differences in several cell structures. Prokaryotic cells lack a nucleus surrounded by a complex nuclear membrane and generally have a single, circular chromosome located in a nucleoid. Eukaryotic cells have a nucleus surrounded by a complex nuclear membrane that contains multiple, rod-shaped chromosomes. 16. All plant cells and animal cells are eukaryotic. Some microorganisms are composed of prokaryotic cells, whereas others are composed of eukaryotic cells. Prokaryotic microorganisms are classified within the domains archaea and bacteria, whereas eukaryotic organisms are classified within the domain eukarya. The structures inside a cell are analogous to the organs inside a human body, with unique structures suited to specific functions. Some of the structures found in prokaryotic cells are similar to those found in some eukaryotic cells, others are unique to prokaryotes. Although there are some exceptions, eukaryotic cells tend to be larger than prokaryotic cells. The comparatively larger size of eukaryotic cells dictates the need to compartmentalize various chemical processes within different areas of the cell, using complex membrane-bound organelles. In contrast, prokaryotic cells generally lack membrane-bound organelles, however, they often contain inclusions that compartmentalize their cytoplasm. Figure 3.12 illustrates structures typically associated with prokaryotic cells. These structures are described in more detail in the next section. Figure 3.12 A typical prokaryotic cell contains a cell membrane, chromosomal DNA that is concentrated in a nucleoid, ribosomes, and a cell wall. Some prokaryotic cells may also possess flagella, hyli, fibri, and capsules. Common cell morphologies and arrangements. Individual cells of a particular prokaryotic organism are typically similar in shape or cell morphology. Although thousands of prokaryotic organisms have been identified, only a handful of cell morphologies are commonly seen microscopically. Figure 3.13 names and illustrates cell morphologies commonly found in prokaryotic cells. In addition to cellular shape, prokaryotic cells of the same species may group together in certain distinctive arrangements depending on the plane of cell division. Some common arrangements are shown in Figure 3.14. Figure 3.13, Credit Coccus Micrograph, modification of work by Janice Haney-Carr, Centers for Disease Control and Prevention. Credit Coccobacillus Micrograph, modification of work by Janice Carr, Centers for Disease Control and Prevention. Credit Spirochete Micrograph, modification of work by Centers. Figure 3.14. In most prokaryotic cells, morphology is maintained by the cell wall in combination with cytoskeletal elements. The cell wall is a structure found in most prokaryotes and some eukaryotes. It envelopes the cell membrane, protecting the cell from changes in osmotic pressure. Figure 3.15. Osmotic pressure occurs because of differences in the concentration of solutes on opposing sides of a semi-permeable membrane. Water is able to pass through a semi-permeable membrane, but solutes, dissolved molecules like salts, sugars, and other compounds, cannot. When the concentration of solutes is greater on one side of the membrane, water diffuses across the membrane from the side with the lower concentration, more water, to the side with the higher concentration, less water, until the concentrations on both sides become equal. This diffusion of water is called osmosis, and it can cause extreme osmotic pressure on a cell when its external environment changes. The external environment of a cell can be described as an isotonic, hypertonic, or hypotonic medium. In an isotonic medium, the solute concentrations inside and outside the cell are approximately equal, so there is no net movement of water across the cell membrane. In a hypertonic medium, the solute concentration outside the cell exceeds that inside the cell, so water diffuses out of the cell and into the external medium. In a hypotonic medium, the solute concentration inside the cell exceeds that outside of the cell, so water will move by osmosis into the cell. This causes the cell to swell and potentially lyse. burst. The degree to which a particular cell is able to withstand changes in osmotic pressure is called tonicity. Cells that have a cell wall are better able to withstand subtle changes in osmotic pressure and maintain their shape. In hypertonic environments, cells that lack a cell wall can become dehydrated, causing crenation or shriveling of the cell. The plasma membrane contracts and appears scalloped or notched, figure 3.15. By contrast, cells that possess a cell wall undergo plasmolysis rather than crenation. In plasmolysis, the plasma membrane contracts and detaches from the cell wall, and there is a decrease in interior volume, but the cell wall remains intact, thus allowing the cell to maintain some shape and integrity for a period of time, figure 3.16. Likewise, cells that lack a cell wall are more prone to lysis in hypotonic environments. The presence of a cell wall allows the cell to maintain its shape and integrity for a longer time before lysing, figure 3.16. Figure 3.15 in cells that lack a cell wall, changes in osmotic pressure can lead to crenation in hypertonic environments or cell lysis in hypotonic environments. Figure 3.16 in prokaryotic cells, the cell wall provides some protection against changes in osmotic pressure, allowing it to maintain its shape longer. The cell membrane is typically attached to the cell wall in an isotonic medium, left. In a hypertonic medium, the cell membrane detaches from the cell wall and contracts, plasmolysis, as water leaves the cell. In a hypotonic medium, right, the cell wall prevents the cell membrane from expanding to the point of bursting, although lysis will eventually occur if too much water is absorbed. Check your understanding. Explain the difference between cell morphology and arrangement. What advantages do cell walls provide prokaryotic cells? The nucleoid. All cellular life has a DNA genome organized into one or more chromosomes. Prokaryotic chromosomes are typically circular, haploid, unpaired, and not bound by a complex nuclear membrane. Prokaryotic DNA and DNA-associated proteins are concentrated within the nucleoid region of the cell, figure 3.17. In general, prokaryotic DNA interacts with nucleoid-associated proteins, NAPs, that assist in the organization and packaging of the chromosome. In bacteria, NAPs function similar to histones, which are the DNA organizing proteins found in eukaryotic cells. In archaea, the nucleoid is organized by either NAPs or histone-like DNA organizing proteins. Figure 3.17. The nucleoid region, the area enclosed by the green dashed line, is a condensed area of DNA found within prokaryotic cells. Because of the density of the area, it does not readily stain and appears lighter in color when viewed with a transmission electron microscope. Plasmids. Prokaryotic cells may also contain extrachromosomal DNA, or DNA that is not part of the chromosome. This extrachromosomal DNA is found in plasmids, which are small, circular, double-stranded DNA molecules. Cells that have plasmids often have hundreds of them within a single cell. Plasmids are more commonly found in bacteria. However, plasmids have been found in archaea and eukaryotic organisms. Plasmids often carry genes that confer advantageous traits such as antibiotic resistance, thus, they are important to the survival of the organism. We will discuss plasmids in more detail in mechanisms of microbial genetics. Ribosomes. All cellular life synthesizes proteins, and organisms in all three domains of life possess ribosomes, structures responsible for protein synthesis. However, ribosomes in each of the three domains are structurally different. Ribosomes, themselves, are constructed from proteins, along with ribosomal RNA, or RNA. Prokaryotic ribosomes are found in the cytoplasm. They are called 70S ribosomes because they have a size of 70S, figure 3.18, whereas eukaryotic cytoplasmic ribosomes have a size of 80S. The S stands for Svedberg unit, a measure of sedimentation in an ultracentrifuge, which is based on size, shape, and surface qualities of the structure being analyzed. Although they are the same size, bacterial and archaeal ribosomes have different proteins and rRNA molecules, and the archaeal versions are more similar to their eukaryotic counterparts than to those found in bacteria. Figure 3.18. Prokaryotic ribosomes, 70S, are composed of two subunits, the 30S, small subunit, and the 50S, large subunit, each of which are composed of protein and rRNA components. Inclusions. As single-celled organisms living in unstable environments, some prokaryotic cells have the ability to store excess nutrients which make structures called inclusions. Storing nutrients in a polymerized form is advantageous because it reduces the buildup of osmotic pressure that occurs as a cell accumulates solutes. Various types of inclusions store glycogen in starches, which contain carbon that cells can access for energy. Volutin granules, also called metachromatic granules because of their staining characteristics, are inclusions that store polymerized inorganic phosphate that can be used in metabolism and assist in the formation of biofilms. Microbes known to contain volutin granules include the Archaea methanosarcina, the bacterium Corynebacterium diphtheria, and the unicellular eukaryotic alga chlamydomonas. Sulfur granules, another type of inclusion, are found in sulfur bacteria of the genus Thiobacillus. These granules store elemental sulfur, which the bacteria use for metabolism. Occasionally, certain types of inclusions are surrounded by a phospholipid monolayer embedded with protein. Polyhydroxybutyrate, PHB, which can be produced by species of Bacillus and Pseudomonas, is an example of an inclusion that displays this type of monolayer structure. Industrially, PHB has also been used as a source of biodegradable polymers for bioplastics. Several different types of inclusions are shown in Figure 3.19. Figure 3.19 prokaryotic cells may have various types of inclusions. A transmission electron micrograph of polyhydroxybutyrate